All right. Good morning, everyone. Jason, can you see me? I do not see me.
Yep, we can see you. So should
Okay.
be going good.
Good enough. All right. Good morning and welcome, everybody, to the second day of the 2021 Idaho Bean School. The Bean School is a joint project between University of Idaho Extension and the Idaho Bean Commission. And just quickly, I would like to thank a few people for helping put this on. Um, Certainly my colleague, Jason Thomas in Minidoka County. He is the technical person behind this, as some of you have been madly chatting and emailing with him about credits, and we'll talk about that here in just a minute. Also, Jerry Neufeld over in Canyon County has helped as well. And then Andy Wolf-Wybe, who is the executive director with the Bean Commission, who has done a lot of the behind the scenes work in marketing this program and whatnot. So we hope that you enjoy the program today. And just a few housekeeping things quickly before we get started. As far as the credits, I saw in the chat there was a question: if I registered yesterday, do I have to register today? Yes, you do. Yesterday was a different session for Department of Ag than today, so they are not the same program. They're two different programs, and so we need to make sure that you were present at both of those. More on that here in just a second. We will run a practice poll question here in just a second to make sure that the technology is working and that you can answer the question. Okay, Jason is putting in the chat there right now that if you are applying for credits, please fill out the survey so that we have your full name and your license number. That's the only way that we have to match your attendance up with this program once we're done today. So Jason, at this point, why don't you run the practice poll and let's make sure that the technology is working. When he runs this poll, I don't care if you answer it right or wrong. We just need to know that you were in attendance. If you are not taking the program today for credit, please don't answer the poll. It just makes a lot more data that we have to dig through to try and match people's time and their name. So we would appreciate if you are not needing credit, do not answer the polls, please. Also, panelists, you have the ability to close the poll. Please don't do that. We'll let Jason handle the poll. We had a little bit of problem with that yesterday. So panelists, I don't think the panelists can answer the poll, but we know that you're here. And so at any rate, with that, all right, Jason, it looks like we have 81. So go ahead and let's close that out. All right, now, don't wig out about the technology. If you did not see the question, or you couldn't answer the question, or you're not sure that you answered the question, here's what I need you to do. When Jason runs the poll questions, I will say the time is, and I'll give you the time. Write that down. At the end of the program today, you'll have 3 timestamps. Send to jerome@uidaho.edu, and I will put that in the chat several times. Those 3 timestamps, your name and your license number, by 2 o'clock this afternoon. Okay, if it's after 2 o'clock, I won't accept it. By 2 o'clock this afternoon, those 3 timestamps, your name, your license number, if you're worried about the technology. That way we will know that you were here and present. Okay, so the first presentation today is for great information. The second presentation is also for great information, but it is also for credit. So that's the one that you'll need to worry about if you're concerned about credit. Now, the Bean Commission was very generous, and in the past we like to do door prizes, and we are going to do door prizes again today. We have 4 door prizes. We have 1 gift card for $25, 2 for $50, and 1 for $100. So we'll do the first one at $25, second one at $50, third one at $50, fourth one at $100. You have your choice for one at either D&B, Tractor Supply, or Cabela's. And Jason will give you information when we get to that point about what you need to do if you are the lucky winner with that. So with that, I think, Jason, is there anything that I missed on the questions or the door prizes?
Nope, I think we are good to go.
All righty. So with that, we will get started right into the program today. It is my pleasure to introduce Jackie Jameson. Jackie is a soil health extension instructor in a partnership position with the University of Idaho and the NRCS based out of Twin Falls. Her work is largely in education and outreach, and one of her primary goals is to create a platform to encourage knowledge sharing and innovation around soil health. This year, she focused on bringing soil health programming to the larger community through virtual field days, and hopefully several of you saw those. A video and discussion format for sharing producer stories and expertise. So today, Jackie is going to take us on a little virtual field tour through some projects that have taken place in the Magic Valley. So with that, good morning, Jackie.
Good morning, and thank you for the introduction, Steve. So just a bit of an overview of my presentation today. It's a bit of a hybrid format. We're going to be starting with a video showcasing a couple of producers in the Magic Valley who were using direct harvest for beans this year. Then we'll move into a discussion, question and answer. We're lucky to have both of those producers here with us today as panelists, so they'll be available to answer questions. Then we'll go directly into our next video, which showcases an organic farm's use of peas, winter peas, as a cover crop in their bean rotation. I'll follow that up with a very short data presentation, and then again, open up to discussion and question and answer. So I guess without much further ado, why don't we get started with a direct harvest bean video?
So my name is Lance Griff, and I farm out here on the Salmon Track south of Filer and Twin Falls. And I direct harvested some beans this year, some black beans, stand-up variety. And this was the header that I used. It's a MacDon D60 dual drive header. And so what I basically did was I planted a cover crop of wheat and barley mix this spring on a field that I had corn stalks in last year. I guess it was thrashing corn, and then baled the corn stalks off, and then came in and just worked the ground once last fall and once this spring. And then I just drilled a cover crop mix of wheat and barley, and then the cover crop came up and I watered a few times just to keep it going. And then I sprayed it out to terminate the cover crop, and then I just direct seeded my beans into the cover crop. So I just used a 22-inch spaced planter and just direct seeded the beans in. And then by doing that, then I didn't have to come back in and cultivate or dike, dam or dike, because it's under pivot irrigation. So I was able to just water the crop basically. I used a burned down herbicide, and also and then I did a post-emerge herbicide also to get any escape weeds later on. And then I didn't have to dam or dike, didn't have to cultivate or anything. And then I was able to just come back in, and that way I could leave the field level and smooth. One thing I probably would have done differently is after I planted it, I would have been nice to come in and roll the field to push down any stones or rocks, things like that. Just because it just helps with harvest. You know, you're not breaking sickle sections and things like that as much. So when the beans were ripe or getting ripe, then I went out and desiccated them to continue the drying process and kill any green weeds that were growing. And then waited about 10 days, and then went in and just this header just fits right on my combine, and was able to just go in and direct harvest them. This is a dual drive sickle head. It drives the sickle from both ends, which helps a lot. One thing, it's a draper head. You can probably see that, but the drapers really help delivering the material to the center of the header. And one nice thing about these MacDon headers is the reel to sickle relationship. Is that you can adjust these, fine-tune them to where you're just bringing the crop in right on those drapers really smoothly, and you can adjust the reel to the desired position you want it, and it really does a great job delivering that crop onto the draper and to the center, and then up ultimately into the combine. So it really did a great job. I was really happy with how it did. I had swathed beans before using a swather, but I'd never direct harvested them like this with a combine. So I had an idea how the header would do, but where the material was completely dry, or mostly dry, it did a great job. And I would say that I don't have any more harvest loss doing it this way than if I had done it the old way we used to do it with the one-step pickets. I don't think I even would have tried this without a draper head. We had enough experience with it. We've had this draper for quite a few years now, and it just does such a great job. And we've used it in different applications, in different things. We swathe wheat stubble with it. We've swathed beans with it on a swather, and so we knew what it could do, and so we were confident we could do a good job with it when the beans were dry and ready to harvest. I would say that the architecture of the bean makes a big difference. I don't think I would have tried this on a bean that is real viney, that lays low, that lays its pods on the ground. But I had grown this black bean variety before, and it held its pods off the ground very well. And so I knew that if it did that again and stood up like it did the previous year that I had experience with, that I would be able to harvest them easily with this. And it did a great job. And even in areas where the soil was not as good, the beans were smaller, they didn't get as tall, it still did a very good job, a respectable job, and just does even better job when you get to the areas where the beans are taller and they're just there's more foliage there to help get onto the headers. Yeah. And one of the good things about being able to use direct harvest methods with this header is that my combine, I can spread all that material out evenly. Whereas before, if I come in and one-step them and put them in windrows that way, then my other combine, my older machines don't have as good of a chaff and residue management system on it. Whereas my newer one, I can spread the whole width of the combine header. So that was a big advantage. Now I can just go back in and no-till corn or whatever crop I want to, because the residue is spread evenly. I don't even have to go out there and harrow it or anything. It's ready to go. It saves time and money, because then I don't have to go out there and spend time and money harrowing it, or oh, this is really thick, I got to go out there and work the ground. It's already done. It's just I can go plant into it, whatever I'm doing. Yeah, we've been trying to kind of figure out how we can use the beans in a rotation with no-till and improving soil health. So it's been we're trying to fit beans into that whole system.
So when this year we direct harvested, I just went into a cover that was terminated, and with my no-till drill with the coulter cart, and so the coulter's running in front of the broadcast seeder. Yeah, this was on a pivot, planted into a cover crop from the previous year, which was stubble. It came out of barley. So I didn't have any tillage before I planted. We did spray more, but I disturbed the soil less. I didn't have any problems putting on water because I didn't have any runoff. Used to have all the old material laying on top and quite a bit of residue on top. So I didn't have to worry about any water running off. It was taking more water. Soil health is a key to helping this process work. Well, this is just saving another trip through the field. The less passes you make through the field, the more money you're going to put in your pocket. This is a flex header, so it'll flex versus the rigid, where it would just plow through some stuff. We did have a little bit of problems with some residue hanging up from my old chaffer rows. So this year I've got another spreader on the back of my combine, hoping to eliminate a lot of those problems. So I have a 2166 case combine with the field tracker on it. This is a 30-foot header. I also have a rigid 25-foot header that I tried one year. We put some lifters on it. I don't have the finger reels. Really in ideal conditions. You don't need to worry too much about the reel. It self-feeds. If you're in good heavy beans, it self-feeds in real nice, and you don't have that loss. You have more loss when the beans are a little thinner. That was my observation this year. So this 30-foot header kept the combine pretty full. I didn't have to worry about keeping the speed up, even though I was thrashing at probably 3 miles an hour plus. It just seemed to work pretty easy. Other thing about this header in my combine, this is called a field tracker. I have the field tracker on my combine and the field tracker on the header. So the header talks to the combine, it senses the down pressure, floats real nice, and it laterally adjusts for any differences. So it's able to hug the ground a little bit better, and I don't have to worry so much about the position of the header and where it's at relative to the ground. So for direct harvesting, we want the ground as smooth as possible. Where we've been doing some cover crop and no-till, that has smoothed up the ground a little bit, pretty considerable. I was worried about the rodents and the gopher mounds. That didn't seem to be an issue this year with this header. Haven't had to worry too much about rocks. That's the other great thing about the no-till, and if you're not going in and ripping or plowing anything, you're not turning up those rocks. So direct harvest loss, rod weeding versus direct header. The rod weeder tends to bury the beans that you may shatter, so you don't see that loss out on the ground. With the direct header, you see more of those beans laying on top of the ground, so it may look worse than the other. We all know that we lose some when we go in there, cut the beans when they're fairly dry. It just all depends on the year. If you cut the beans a little green, then you're not losing as many with the rod weeder. So who knows? That's up for debate.
Okay, Jackie, did you want to take some questions now?
Yeah, thank you. So a couple of questions were answered already by Lance in the question and answer, but I did want to bring them to the table because I think a few of them at least are of interest to probably more than a few people. So as I read those out and read Lance's answers out, please feel free to continue to add questions into the Q&A or the chat, and we'll address them as they come up. So one of the first questions was, how was yield compared to previous year? And Lance said it was similar, but would have been better, except the beans got frozen this year around the 10th of September, which I'm sure a bunch of these folks have seen as well, and hurt yield. Lance, did you want to talk a little bit more to that?
Yeah, sure. So I feel like the beans grew well and they looked good. It's always something where trying something new, you're not sure exactly how much fertilizer to put on, how much carbon penalty is there going to be with all that residue out there. Should I put more nitrogen on to take care of that, to help break down that to volunteer barley and wheat that was there? And so I think in the future I would do that. I put more nitrogen on to kind of take care of that residue that was out there. But it was hard to compare yields because we got that frost in September, early September, and the beans weren't finished yet. So we lost a lot of those smaller beans that were still finishing, and the beans just basically shut down. So it was hard to compare, but I feel like the yield would have been, it wasn't great this year, but it would have been quite a bit better without that frost.
Thank you. Todd, did you have anything to add to that? I wasn't sure if you had any fields for direct comparison this year, if you had some that were direct harvested and some that were not direct harvested, or if you have any additional insights to add there.
I feel they did pretty well for this year. I did lose some because of the old chaffer row from last year. That's where the green beans were. So we harvested them, and there was some that were higher moisture. By the time they milled them, they came out. So it averaged 16 bags per acre. So without all the extra steps in there, I think it was a win.
Thank you both. Another question about the post-emerge herbicide that was used. Sounds like Lance used Varisto and grass control herbicide to get some of the remaining volunteer barley crop out. And then the next question was, could you try this behind corn stubble without baling the stalks in the fall? Lance, did you want to elaborate a bit on what you wrote there? Because it sounds like you do have some experience with that.
Yeah, so a couple years ago we tried that, and we just came in. We just harvested corn. It was grain corn, so we just left all the stubble there. We didn't disc it, we didn't shred it, we didn't bale it, we didn't do anything to it. So then the next spring, as an experiment, we decided we tried to plant beans into it. Some of you may have seen the video from a couple years ago. So we just planted directly into that corn residue. Planter didn't have any problems going through it because by then the stalks were dry and brittle, and just planted right through it. And then at that point, I wasn't comfortable yet just going out and stumping the beans with the combine, desiccating them and stumping them. So we just swathed them, and the header did a great job on that in that situation too. So the extra corn stubble in the beans when we went to swath it didn't cause us any problems. The header went right through it, didn't give us any issues. So I'm confident that you could do that right, doing the same thing, just cutting out that swathing step and instead just spraying and desiccating those beans. And then going in like I did, showed in this just most recent video. So yeah, I think that could be easily done that way.
Thank you. Another question on how many years have you done these practices? Maybe Todd, we'll start with you on that one.
Yeah, this is in my third year of trying this. So it seems to be getting better and better every year.
Excellent.
Yeah, and for me, I would say in that 3 to 4 year range, and every year I seem like I learn something new and try to incorporate it into the system. So I'd say I'm similar with what Todd's been doing.
And the last question we have open right now is, do you feel the cover crop helped with weed control? Both of you did use a cover crop prior to planting the beans. Do you feel like that cover crop helped with weed control in your beans this year? Lance, how about we start with you here?
Yes, I do feel like it helped with weed control. Excuse me. Yeah, I just seemed like there wasn't that many weeds out there. I went ahead and sprayed the Varisto and then the clethodim to get any of the volunteer wheat and barley cover crop that had escaped. but I didn't have a big weed mess. People came out and saw the field, and I don't, I just, excuse me, I feel like there just weren't that many weeds out there. And yeah, I really think it did a good job of holding the weeds back.
Thank you. Todd, what were your observations this year?
Yeah, I think the cover crop helps hold the weeds back, and I think it's a positive thing in many different levels.
Excellent. And the last question as of now is how do you address fertilizer? And I'm assuming this is accounting for the residue in the field as well as the different approach to bean management. Todd, did you want to start on this one?
Yeah, I put on a little bit of fertilizer just to help break down some of the residue. I know in some of my other no-till fields, the nitrogen has been more available later on in the season. It seems like as things start to break down. I'm still learning.
Yeah, and I would echo what Todd says. It's a learning process every year, it seems like, with switching over to something new. But on my planter, I've got a 2x liquid. I just go liquid on my planter. So I'm putting on 32% nitrogen 2x2, except I just dribble it on the surface on both sides of the row. So that's how I'm getting my nitrogen. And then I put it in biologicals and some starter fertilizer in furrow. And that's how I'm doing my fertilizer for the beans.
Another good question on here about water. I'm curious your takes, and the question is how much more water did it take? And I'm curious how, if you could both compare water use with this type of management versus maybe more conventional bean management.
So I guess for me, I used quite a bit less water using the cover crop and not having to dam or dike. And I think a big portion, kind of a twofold reason why, is that for one, the soil was fairly well armored with that residue out there. And so I wasn't getting that evaporation in the heat of the day in the summers. So that was one benefit. And also, it just seems like the water, well, I know the water infiltrates so much better in that system, in the cover crop system. And so the water that we're applying is going in and being held there rather than putting it on and using a dam or diker to kind of hold, use those pockets of water that, sit there, and some can evaporate out. And so the residue just really helps hold that moisture better. And then I can put more water down with each revolution of my pivot. And so I'm being more efficient with my pivot also. So I can slow my pivots way down and have them go a lot slower. So then it's more efficient, just more efficient use of water. So I would say I probably used 2 to 3 inches less water this year. And I was always surprised when I'd go out there to dig with my shovel, how I always expected it to be drier, and it seemed like it always was wetter than I expected it. And so I just ended up turning my pivot off a lot more frequently than I normally would on a normal summer. So I use less water.
Todd, did you have anything to add to that?
Yeah, I probably used a little more water earlier on. And partial of that was I didn't have as good a seed to soil contact in some areas. So I was using the water as a tool to help get those poorer areas to sprout and up. But later on, my usage was probably down. I see Bill had a question about mold. In all the fields that I've had cover crop in, and well, pretty much all my fields this year anyways, I didn't have much mold problems or any mold problems.
And Lance, I remember you experiencing a similar situation with less mold pressure, at least this year. Is that correct?
Yes, yeah, I would concur with what Todd said, and that every year that I've done cover crops, ahead of the beans, I didn't have mold, even in situations where I thought, I've got a whole bunch of residue here. I kind of expected there to be some mold, but I think we've kind of talked about before, and Bill and others have theorized why that is, and we kind of came up with the thought that maybe it's because you're not getting that splash, that water splash up onto the plant when it hits the bare soil, and is spreading the sclerotia up onto the the plant itself, where you've got that residue there, keeps that raindrop from splashing that soil up on the plant.
Excellent. Thank you both. And one last question I'd like to answer or address before we move on to our next video, just in the nature of time. What are your top 3, or maybe I'll get 1 or 2 from each of you, pieces of advice to someone wanting to switch from conventional bean harvest to direct bean harvest? Maybe Todd, if you could start with 1, and then we'll go to Lance for 1, and see if we can pull together 3 here.
I would say the main advantage is you're not laying those beans down on the ground. If you happen to have a rainstorm and your beans are still standing, it's not a big deal. And it's another tool for you to use. And also your beans don't get blown. I've had some beans in windrows this year that got blown. It's a big step. It's tough because it's new, but I think it's worth it.
Yeah, I agree with Todd. It's like he said, it's different. It's, kind of new. It's, I'd say try one step at a time. If you're uncomfortable with having your beans stumped with a combine and doing all that, find somebody that can come in and swath them. But I would say try to find a bean that stands up decently for you. One of the issues that I have is being out on the salmon track and being higher elevation than other areas, that we run into a shorter season. And so the issue for me is finding a bean that stands up well enough, that's a short enough season. And that's kind of been the challenge for me. And so if you can find a bean that you're happy with, that stands up well and holds its pods off the ground, that makes a huge difference, in my opinion, on how well the header works as a system. So if you can find a bean that stands up well, and maybe start having somebody swat them for you and see if you like that. If you like that, then maybe move from, do that step, go another step the next year, start on however many acres you feel comfortable with.
Thank you. Hearing some important words of wisdom there of kind of starting incrementally, and using what resources you have available to you, being careful about variety selection, and knowing from experience that it works, and giving it a try, and outweighing, or benefits outweighing some of the costs or risks of that. So thank you very much, Lance and Todd, both for sharing your insight and input. And I hope that if questions do keep funneling in about this topic, please continue to ask in the question and answer, and hopefully Todd and Lance can continue to answer those in the text. But in the nature of time, I would love to move on to the next video, which showcases the use of Austrian winter peas in an organic crop rotation that includes beans. So Jason, if we could get that up and going, that would be wonderful.
We are on Joslin Farms today. This is a field we've been farming for about, the last 7 years or so. We bought it from a neighbor, and we've come in and installed our organic farming program, you could say. Our rotation would probably be definitely is based and centered around an alfalfa crop. So we'll put alfalfa in, leave it in for, probably between 3 to 5 years, but 5 years being really long because we're generally pretty excited to get back to a row crop situation. Here where we have wheel lines, the row crop would definitely be beans, just like we have here, but it goes the same as far as time to take the hay out. If it's under pivot, we'll take the hay out, do our bean crop, then it'll go to probably barley. And so then we'll put the barley crop in either in the spring, or if we get a good year, we can get the beans off early and get that winter barley in. But anyway, do a barley crop. Then after the barley, what makes barley it's as central almost to the rotation as the alfalfa crop is, it's off early. We get to start our Austrian winter peas that we get in, in the, I guess you could say late August. That's ideal, I suppose, for us. We have put the peas in after the beans come off late in the fall, and they do make it, but they're maybe a little weaker. But yeah, back to the rotation is barley, get our peas in, and then they winter over, come up real strong and healthy in the spring. We let those grow till about May, the middle of May sometime, and then incorporate them into the ground, or we'll chop them off maybe and sell them to the dairy. But we incorporate them into the ground, and those peas have fixed a lot of nitrogen for us and are a nice cover crop. They keep the weeds down really well in the spring. It's kind of like, well, it is exactly suppressing our weed load until we have to incorporate them and expose the ground to the sunlight, to everything, to water, to everything that the peas aren't using up, or covering. So they're a great, I guess, segue into our beans and or a corn crop. And then that comes off late in the fall. We may do peas again in the fall, or a spring barley, and kind of keep that rotation up where we go barley to peas to row crop. And that's what we've been doing. And then as time goes by on those row crops, and the thistle may come in and just get harder to manage and harder to manage, that's when it's time to get back to hay. But generally, hope to get 5-6 years of this row crop barley thing in the middle, and then back to hay. So that's our kind of explanation as far as rotation goes. Right, but this isn't, fluff. We're really growing beans, Fall barley crop in the fall of, let's say, 17. It sits there all through 18. Then you're actually tilling the peas in 19. So you're doing so much no-till, and you go out there and you feel the dirt, and it's that cakey, crumbly, balled up, little nice soil texture, and it's just really great. And then you got worms everywhere. There's worms in everything. When it sits that long, when it can sit for a year and a half, pretty much with no tillage at all, only stick the peas in, only that, it's really nice. It really makes a nice soil profile. And then like right now, if we went and pulled up one of these bigger root balls out of one of these weeds, that's a nice root ball, there's gonna be 10 or 12 worms in it. There's a bunch. I pulled some up in the cornfield the other day, and you pull out a big nasty weed root ball, but there's all these worms here, there's 2 or 3 there. I mean, they're real happy in it. So the peas fit into this rotation so well because this is an organic farm. I probably mentioned that already, but our biggest constraints, our biggest limiting factors are weed load and fertility. And the peas, as I said before, they shade the ground, cover the ground so well. As soon as they get going in the spring, as soon as they stool out a little bit, we're planting about between 100 or between 80 and 110, kind of give or take. It doesn't have to be an exact science. We're spreading peas on with a spreader cart, and I think we just kind of said, yeah, that looks good. So we'll incorporate that. We'll disc those in, and you know, to what, and then we'll water and get them up. But that pea, as soon as it starts stooling in the spring, just covers some ground, just looks great, and covers up all the weed, you know, fights the weeds back. And so that's just huge for us with the organic row crop. And then also adds the fertility of those little nitrogen nodules on the root system, which we obviously don't have at the moment, but boy, we sure looked at them close this spring when we were tilling them in, and they were just nice long roots that had the little pink bags on them, and there's our nitrogen. So it's a great deal. And I think it's generally assumed that beans don't need nitrogen because they're a legume, but they need some help when you're farming organically and you don't have much you can offer them.
They're kind of the bridge.
And the peas are the bridge crop. There you go. Or yeah, and they're great in that they winter over. They're in the middle. We're not really trying to do anything else. It's not like it's a 100% win-win. Peas go in the fall after a barley crop that's been just good part of a rotation, and then off they come in the spring, you know. In about the beginning to middle of May. The best routine we have for handling the peas is use the cornstalk whipper and then disc them. We have a high-speed disc, and it should be able to incorporate a pea in a couple passes, actually, as I see it. I mean, where we kind of got this year with dealing with these big, robust peas was, and I think I even mentioned it to Jackie, was really the best time to incorporate these and start this process of killing the peas off is when they're the perfect feed for a cow. They're at their best, get them turned in because they're not huge. You can really manage them easy when they're just not so big. And you're incorporating the best you can put in, you know, that they're at their peak. And they're only yay tall, yay big, I guess. So whip them, disc them a couple times, call it good. Let it sit. You know, I believe that's another thing is don't get too antsy because you've still got a while until you're gonna actually plant beans, or if it's corn, that's another story. Yeah, you wanna have that in at the end of May or something. But yeah, let them sit for a second, because if you let them sit, that's another great thing about those the pea is, there's so much water to them. I think once you incorporate them, the ground starts digesting them really quick, you know. It's just a nice fast breakdown and they're gone. That nitrogen that's fixed by the peas, I don't believe the beans are using it all up. There is a carryover nitrogen that the barley gets to take advantage of, and it just helps make that barley crop a little better. We are putting some chicken compost and fish emulsion in at the planter with that barley seeding, but shoot, we're happy with our yields on the barley, so what we're doing there is definitely working as well. That crop is always in the ground. You know, we try as best we can to keep something going. The bean crop, after the fact, it's just a little too late to get much started unless you get them out early, you know. And I don't know, this particular year as we're talking, I know of a field that we've got, it's turning yellow. A neighbor has cut beans. I'd be trying to get peas in there, you know, so. What we'll do after these beans come out is really something we can really only answer when they're gone. I mean, these are reds. I expect them to be a later, you know, harvest. Like I said, we have put peas in I don't know, it might have even been late October. And you just have to hope they get a little moisture to pop up. They definitely don't produce what they would if you get them in early, but it's a game time decision, really.
Thank you for the assistance, Jason. Before we get into direct questions with Alex, I'm going to share a little bit of data from a kind of mini project that we did this year. Bear with me here while I share my screen. Share screen, here we go. Jason, are you able to enable?
Yep, you have permission now. I just said it.
Okay, perfect. All right, can everyone see that? We good?
Yes.
Yes.
Excellent. So as Steve mentioned, my name is Jackie Jameson. I'm the Soil Health Extension Instructor with the University of Idaho and Natural Resources Conservation Service. This mini project that I'm about to talk about, nitrogen dynamics after a pea cover crop and an organic rotation, was kind of initiated and supported in collaboration with the Twin Falls Soil and Water Conservation District. Upon seeing the successes of the crop rotation at Jocelyn Farms, we decided to look a little bit more closely into what was going on with nitrogen in this field in an effort to better understand the soil health impacts of a winter pea cover crop. So I want to throw out here right now that I call this a mini study because it includes only one season of data on a single farm. There aren't replicates. It's more of an observational study, and it, as many studies like this do, brings more questions to the table, I think, than answers, which is really exciting. It's not intended to be comprehensive or to tell the whole story, but there are some interesting points to note that I'll bring out, and even more interesting questions that arose. So I plan to briefly share what we found as far as data goes, and then kind of open it up to further discussion, both about the video and about this little data set here. So a bit of an overview of this field's history to kind of recap from that video. The fields that we were looking at were planted to Austrian winter peas at a rate of about 100 pounds per acre in August of 2019, following a barley crop. These peas were then mowed and incorporated with a disc in early to mid-May of this past growing season. And then beans were planted about 2 weeks later in late May. Wait, why is this not working? Sorry about that. I'm not sure why this is not working here. There we go. So these are the peas after mowing, peas after incorporation, and the beans growing in the end of August. And all of these clips you could kind of see in the video as well. Samples were taken from 0 to 6 inches, and I want to throw out here that these samples were not intended to be used for fertility recommendations, but rather to look at nitrogen dynamics in the top of the soil profile, where it would be most impacted by the peas. Our soil sampling protocol was consistent across the 3 time points, and it was really used to look at some of the soil health parameters that might be able to help explain some of the successes experienced by the Jocelyn. Soil samples from an adjacent field were included to fill in data gaps and are recorded as a star in all graphs. And I'll also point those out so you can be aware, but they really add some interesting points to this. Now, this adjacent field was managed very similarly to the one in question and shown in the video. It was just in that rotation 1 year previously. So 1 year ago, it was in the peas beans rotation. And then the Haney test was used for all metrics shown here, except what's in the first graph. The first graph shows a traditional soil test nitrate in which traditional KCl extraction methods were used. So showing this first graph here, this, as I mentioned, is showing traditional soil test nitrate for the adjacent field. You'll notice on the x-axis on the bottom here that this is a pretty long time period, and these samples are pretty widely spread apart. So we're talking about June of 2019, so not this past season, but the season before, to June of 20. That's a year in between these samples. And late late fall, November of this past year as well in this field. So again, this is for the adjacent field, just for reference. General trends showing after the peas were incorporated early in the bean growing season, soil test nitrate is still pretty high. And then into the spring barley, the soil test nitrate, once that barley was growing, is relatively low. But what's really interesting, and I think this is a question that came out of this year's mini study as well, is what's happening in between here, in between the beans and the barley. What's happening to nitrogen in there? How is the barley, when it's planted in early spring, how is that experiencing nitrogen in the soil? So what we're going to be looking at for the rest of these graphs are all Haney test results. So the Haney test, for those who aren't familiar, is a test that uses water and a weak acid extractants in order to better assess or more closely assess the soil environment that plant roots are experiencing. So this H3A extractable inorganic N includes both nitrate and ammonium, so plant available nitrogen in mineral form. And what we're seeing here reflects 3 different time points: early in the season, right at planting, 3 months later in August, so pretty late summer, and then 3 months after that in November. So again, these time points are pretty far spread apart, especially for something that is as sensitive to management and temporal changes as the Haney test is. And if we only include the data from this one field, these 3 points, we see an initial high point of nitrogen about 2 weeks after pea incorporation in this May 20. This is at bean planting, so 2 weeks after pea incorporation. We see a relatively high point in nitrogen and then a dip in the summer, likely as the beans are using that nitrogen, and then a subsequent increase in later November. This is consistent in organic nitrogen as well, which I'll show in a moment. But what I want to show here is an interesting point. If we include a soil sample from the adjacent field last year, that's a time point from June. So about a month after peas were incorporated, we see a big spike in nitrogen availability. And this is pretty consistent with other studies that show 50 to 75% of nitrogen from leguminous cover crops being available in that 4 to 6 week window after incorporation. So we are, if we include this extra sample, experiencing that similar trend here. But as much as that makes sense, what we're really interested in is these residual effects that happen afterwards, both later in the season and into the following season. Water extractable organic nitrogen is the organic nitrogen that's easily extractable in water and essentially easily mineralized, so turned from organic into nitrate and ammonia that's available for plants to use. And we see a similar trend to here, although it does seem to be increasing a bit more at the end of the season than we saw with the inorganic N. Similarly, we see a peak if we include the data from the adjacent field. And the last graph I want to show here is the organic C to N ratio. This is a number, a calculated number from the Haney test as well, that includes the water extractable organic carbon and the water extractable organic nitrogen in here. So what we see as a general trend is throughout this growing season, organic C to N ratio was in the good range for microbes. So what that means is that nitrogen isn't being tied up because there's too much carbon, and likewise there's not too small amount of a carbon to feed the microbes. So microbes are generally going to be relatively happy in this range of C to N. So with that, I could talk a lot further about this data, but we are limited on time here. I do want to share a special thanks to the Twin Falls Soil and Water Conservation District for their financial support and initiative in this project. And I wanted to throw my contact information up there for those of you who might be interested in getting more information about this. But we're really looking forward to hopefully continuing this mini study into next year to at least get an idea of what those plants in the spring are experiencing as residual effects of the pea cover crop. So not just the first year, but into the second year. So stay tuned for more information on that. So I will stop screen sharing here and we can move into question answers, discussion, etc. Jason, am I already stopped screen sharing? Did you do that for me?
Yes, I did.
Thank you. All right, so I see a few question and answers in here. Jacob asked, what variety of pea are you using? Alex, I can't remember what variety of Austrian winter pea. Did you already answer that?
No, we haven't answered that. I'm
Okay.
trying to think. On the cedar seed, it just says Austrian winter pea.
Okay.
Yeah.
All right.
Okay. We have tried, as I mentioned in that video, we've tried icicle peas, and those didn't turn out very well. They don't stool out quite like the Austrian winter peas do, I guess. They just kind of grow up spindlier.
Gotcha.
So
I do want to put out here, we are going to do probably 2 questions before we move on to the next speaker. And for those who are interested in continuing the Q&A session and discussion, we will have more time for discussion after the next speaker. So stay tuned and stay online for that. One more question for you, Alex, from Lance. Do you worry about volunteer peas in your beans the next year?
We do worry about it, but typically we've had pretty good luck getting the peas knocked down and killed with disking and disc ripping. It really hasn't been a problem. We did have one field this year that had some peas in it, and I think what do we do? The beans had to kind of go a little grade down, those ones at Coiners.
Well, they just, I think, put a little extra effort into the milling because of the size of the peas they mill out.
Yeah.
So, but it hasn't been a problem.
It hasn't been a problem. That's the first time we actually had an issue with it, but the peas generally kill out real easy. The disking, and they're halfway to, compost. So, especially when, as you saw, we're whipping them as green as they are, when they're so full of moisture, you disc them in and they just seem to, they take care of themselves awfully quick. They decompose very quickly. So.
And a similar question, do you see any barley volunteering in the peas if you have peas after barley?
We do have some volunteer barley when we're going from barley into peas, then in the spring, both of them come back. That's also part of the cover crop there, is that volunteer barley contributes what it can. And then in the situations where we've like last spring, we chopped a few fields, and that barley just helps make tons, and they're a nice tender stage, make nice feed. So but then after that, no, we haven't really had any volunteer barley after, you could say, that incorporation that I can think of.
That's right. We're killing them in the vegetative stage. So there's obviously no peas. If there is a volunteer, it's probably from hard seed that didn't germ.
Excellent. Well, thank you, Alex and Carl, so much for your time and input, both in the video and for being here today. Like I said, we'll revisit some of these questions and such later in the programming after the next speaker. But I do want to be respectful of time here and hand it back over to you, Steve.
All right. Thank you, Jackie. Appreciate it. Really interesting videos there. Again, hope the panelists can hang tight after the presentation today if there are some additional questions or discussion that folks would like to have about some of these new practices that we're seeing start to be incorporated here in the Magic Valley. All right, Jason, we are up to door prize time. So for a drawing for a $25 gift card, who is the winner?
All right. Well, we're going to have to make sure that they're actually here because I could not find them in the chat and in the participants. So using registrations, I randomly drew a name and the winner is Stephen Meyerhoff. If you are here, let me know in the chat, or you need to call the Minidoka County Extension Office to confirm that you're actually here. If we don't hear back from you within 5 minutes, either through the chat or calling my office, the phone number here is 208-436-7184. So we need you to call us if you are actually here, Stephen. Otherwise, we will redraw. We're going to do another one too, right, Steve?
Yep, this one is for $50.
And the $50 one goes to Lance Griff. That was totally random, but yeah, one of our panelists. So
Sounds rigged to me.
and he is here, so makes our job easier. So again, reach out to us, Steve, in the chat or call my office. Otherwise, we will do another drawing for that $25 gift card.
Okay, thank you. And Lance, if you would just do a private chat there with Jason and let him know whether D&B, Tractor Supply, or Cabela's gift card you would like. All right, great, and congratulations. And again, at the end of this next presentation, we'll do another drawing for a $50 card and then finally a $100 card. So stick around for the chance to win for that. Okay, our next presenter is Dr. Lindsay DeToit with Washington State University Extension. Jason, before we get started, would you please run the first poll question? This will be the first question that you must answer for credit, and the timestamp is 10:04. So just go ahead and answer that quickly. You don't need to think too hard about the question. This is pass fail. If you showed up, you pass. If you showed up and answered the question. All right, about 5 more seconds. Looks like we've kind of stalled out there. All righty. Thank you, Jason. And again, that timestamp was 10:04. Okay, our next speaker is Dr. Lindsay DeToit. She serves as professor and extension plant pathologist at Washington State University, Mount Vernon, Northwest Washington Research and Extension Center, where she runs a vegetable seed crop pathology program for the past 20 years. She focuses on disease affecting small-seeded vegetable seed crops in the Pacific Northwest. Today, Dr. DeToit will review seed-borne bacterial pathogens of bean that have quarantine significance for bean seed production in the Pacific Northwest USA. The presentation will cover the causal agents, their biology and epidemiology, and management recommendations. In addition, the recent history of outbreaks of these diseases in the Columbia Basin will be examined along with similar looking problems that have been misdiagnosed as bacterial diseases and complexities associated with testing plants and seed for these two pathogens. Dr. DeToit, good morning.
Good morning. Thank you very much. Can you hear me all right?
Yes.
Okay. Am I able to share my screen?
Yes. Jason, does she have screen sharing?
Yes, she does. She should.
You do.
Okay.
All right. So Jason, can you let me know if you are able to see the slides up?
Yep, we can see it, and it looks like you're in presentation mode, so that's perfect.
Excellent. Great. Thank you so much. And appreciate the opportunity to share a little bit about some of the experience we've had with bean bacterial pathogens that are a big concern for seed production in Washington, but also obviously in Idaho and Oregon, being an important seed production region, or the tri-state. There are a number of bacterial pathogens that affect beans. This is a list of 5 that in the literature, if you find out, try to find what bacteria go to beans. And the 4 that I've highlighted in red boxes here are the ones that have become, historically have potentially been an issue in bean seed production, and recently in the last 5 to 10 years are continuing to show up more regularly, certainly more than we would like. Halo blight is probably one of the most well-known, Pseudomonas syringae phaseolicola, and common bacterial blight, which is a Xanthomonas pathogen. Bacterial brown spot, which is very closely related to halo blight. It's just a different pathovar. So it's Pseudomonas syringae instead of Pseudomonas syringae phaseolicola. And then bacterial wilt is a different bacterium, quite different than the other 3. It's Cotobacterium floccumphaciens, and it's a specific pathovar within that, also called floccumphaciens. And this is a disease that is starting to show up more than we would like. It showed up more recently than these other 3, and seems to be increasing in problems. And so we're learning a lot more about this. There's a lot more to be done, and really need to be keeping an eye out for this. So in Washington State, somewhat similar to Idaho, being an important bean seed production region, we have a quarantine that regulates the list of pathogens on this slide. So not only bacterial, but also the fungus, the fungal disease anthracnose, as well as bean common mosaic virus. And again, in 3 red boxes here, I highlighted the 3 pathogens that we have seen show up in bean seed crops since about 2009, sporadically, but just concerning about how much more regularly we're starting to see things each year. So these 3 pathogens that we've been dealing with in Washington, and I know that have been showing up somewhat in bean seed crops in other states in the Northwest. Halo blight and brown spot, as I mentioned, are very closely related. Same genus and species, different pathovar, and then common bacterial blight, Xanthomonas. There's quite a bit of diversity within these. There's known races of the halo blight pathogen. So when breeders are trying to develop resistance, they have to be able to screen against the different races to see if they hold up against all of these races. This pathogen does best at temperatures that are sort of moderate, whereas Xanthomonas tends to do well at warmer temperatures. So that's quite an important distinction. And brown spot as well tends to do well at warmer temperatures. The halo blight gets its name from that yellow halo around the lesions, the spots on the leaves. And that halo is produced as a result of the bacterium releasing this toxin called phaseolitoxin. And that toxin is produced in its highest amounts at these sort of moderate temperatures. When temperatures get warm, that toxin production tends to decrease. And so you can actually have infection and lesions without that halo when it gets particularly warm. The brown spot pathogen also produces a toxin that allows it to cause these symptoms on leaves. So how do you manage? What's sort of the theoretical recommendation for managing these pathogens? And I'd like to start with this so we can proceed into why it is we maybe are starting to see these diseases show up, and in contrast to the recommendations. So we know that these pathogens are seed-borne and very readily seed transmitted. And so contaminated seeds are definitely an important inoculum source. So if you're planting a bean seed crop, making sure that that seed you're using for the bean seed crop is clean is extremely important. When those seeds germinate, the bacteria very quickly colonize the seedlings that are coming out of that seed. And then the bacteria are spread basically mostly by water. So whether it be rain or irrigation, which you have to have for a crop, unless you're doing drip, which is not very common for beans. Furrow irrigation tends to be less favorable, rill irrigation, because you get less splashing. Windborne rain irrigation, windborne soil. These bacteria don't persist very long in soil, but if you've got infected residues in the soil and that soil gets blown with those residues, it can certainly move them around. One of the ways that it can spread in a field is if you have either equipment or field crew or animals moving around when the crop has been wet following an irrigation or rain, and the bacteria oozing on those infected leaves, and people or equipment moving through can potentially move that bacterial ooze that's on the leaves. These bacteria are favored by moisture, including high humidity. Conditions when you go wet and moisture, that's always going to be more conducive. As I said before, halo blight tends to be favored by moderate temperatures. Sorry, these are Celsius, but just know that that's moderate, high 60s, low 70s. The other two do better at warmer temperatures. And another complicated part about this is that just because you don't see symptoms in a field doesn't necessarily mean the bacteria are not there, because there's this delay after the plants get colonized before symptoms start to show. And the halo in the halo blight symptoms can disappear when it gets particularly warm, so above about 78 degrees, 80 degrees. These bacteria survive on infested residues. As I mentioned, they're not truly soil-borne. They'll only persist in soil typically as long as there's infested residues. And that's why residue management becomes particularly important. They also survive on volunteers. We just heard questions to the previous speakers about volunteers from the previous year's crop, volunteer beans or volunteer peas and so on. And there are some other hosts that can get infected, including certain weeds. So I'm going to show you a range of pictures just to get an idea of the differences in symptoms you might see. The halo blight pathogen, when it infects a leaf, the early symptoms are these tiny pinprick sized little dead spots, and we say necrotic, which basically means dead spots. And as they develop, you start to see this clearing zone around it called a halo or chlorotic or yellow halo as the toxin is released and starts to break down those cells. If you turn that leaf over when an infection is just starting, you'll see these small pinprick water-soaked lesions where the bacteria are colonizing the leaf. And then, of course, as infection develops, you get those classic halo, yellow halos around the dead centers where the bacteria initially colonized. That's sort of the classic. And it can get quite severe. These are photos taken by Howard Schwartz, who's retired from Colorado State University, showing some pretty severe outbreaks. And in places like Colorado, where they get these big storms come through, this can be very problematic for these diseases. And this is sort of an even more severe case of halo blight in a field in Colorado. On the pods, you can get these water-soaked spots, again, very similar because the bacteria are breaking down the cells. As the cells break down, they release the contents and you get that water-soaked appearance. And the bacteria, when under moist conditions, can actually ooze out onto the surface of the pod, and you get this sort of dried bacterial ooze, and the seeds become shriveled, discolored, and infected. And here's a range of symptoms on some pods, again, some pretty severe. The bottom picture, you can see the lesions on the pod, which were very severe, have now moved onto you can see this middle seed has some water soaking on the seed where infection has gone into the seed. If we look at the brown spot pathogen, there's some similarities, but also some distinct differences compared to halo blight. You don't get that big halo. You do get a little bit of yellowing or chlorosis around the lesions, but you don't get that distinct big halo. So you get those dead areas on the pods and on the leaves. You get some yellowing around it, but you don't get that very large halo. This is a photo taken by our Washington State Department of Ag folks of a bean seed crop that had an outbreak of brown spot, showing some symptoms on the pods, as well as some fairly extensive symptoms on the leaves. But you can see the distinct lesions on those pods. Here's some additional photos from that field. And you can see the top left picture is not that distinct. It's kind of, there's a lot of things that could start to look like that. But the pod lesions in this photo that Paula Moore took, who's the seed program manager for WSDA, are pretty distinct. Common bacterial blight, early symptoms can look quite a lot like brown spot. Again, you get water soaking where infection takes place. You get the dead tissue with some yellowing around it. So, it could easily be confused with brown spot. These are some more mature lesions. They tend to be pretty irregular. And here's some very severe cases, again, taken by Howard Schwartz in the Colorado area of some severe outbreaks of common bacterial blight where they were doing some research trials. And these are symptoms on the pods, again, that water soaking because of the bacteria breaking down the tissue, the dead spots as they dry out, but not as distinct as large as the brown spot and halo blight. So I think it's time for a poll question. I've been asked to put 2 poll question slides in here. So.
Okay, Jason is running that second poll question. The timestamp is 10:16. Okay, let's give that about 5 more seconds. I think we're about there. Okay.
Great.
Thank you.
Thank you. I'll move on. Let me. Okay, so in Washington State, the bean seed quarantine was implemented in 1968. So going back quite a long time ago, it was implemented at the request of seed growers and seed companies to try and protect against the introduction of these seed-borne bacterial pathogens into areas where bean seed is grown, and obviously to try and keep those areas clean and be able to assure production of healthy seed lots that could be sold around the country and around the world. In 2009, and so that's considerably long time after 1968, 5 fields ended up testing positive on the harvested seeds. So 2 of them tested positive for halo blight, 3 of them tested positive for common blight. All 5 of these bean seed crops, all 5 of them were under center pivot production. Now I know that there are some discrepancies in the Washington and Idaho State Department of Ag rules about whether or not you can produce seed crops under different types of irrigation. But in Washington State, you can use overhead irrigation, but we know that that tends to be highly favorable for these crops. So all of these fields were under overhead irrigation. This is, this was disconcerting, and all 5 of these seed lots had to be destroyed. They couldn't enter into the seed market. So, significant loss for the seed company, and of course, a concern for why these 2 diseases showed up in 5 fields after nothing having been detected since 1968. In 2012, a 17-acre bean seed crop up in the North Columbia Basin, also under center pivot irrigation, showed up with halo blight. You can see the distinct halos on the leaves. And that crop had to be destroyed after confirmation of the diagnosis. In 2014, so in 2013, to my knowledge, we didn't have anything, so that was good. It was a relief. But in 2014, unfortunately, there was a cranberry bean seed crop in the North Columbia Basin. You can see this lower picture. This looks like little sort of UFOs that landed all over the field. There were hot spots throughout the field. And this again was under center pivot irrigation, confirmed by a certified lab, and the crop had to be destroyed. So, now we were in our 3rd year of having to see some crop destruction and losses to both the grower and the seed company, which is disconcerting. In 2015, to my knowledge, we didn't have anything, but in 2016 and 2017, there were at least 5 bean seed crops, all under center pivot irrigation, that ended up testing positive for either halo blight or bacterial brown spot. And what was interesting is that the halo blight strains that came off crops in those 2 years did not actually produce a halo. So they were initially thought to be brown spot, but on further testing, and I'll show you some slides coming up, it was revealed that these were what we call non-toxigenic or atoxigenic strains of the halo blight pathogen. So they don't produce a functional toxin when they grow, and the symptoms can look a lot like brown spot. And so here's some photos, again, of pods with very distinct lesions and some leaves. This is a photo taken by Lyndon Porter, USDA legume pathologist. So pretty distinct lesions and pretty severe outbreak. As I mentioned, this sort of non-toxigenic strains of the halo blight pathogen. So Pseudomonas syringae phaseolicola, the halo blight pathogen I mentioned, causes these halos because it has this toxin called phaseola toxin. The strains of the bacterium that have a functional toxin gene are called Tox plus because they have the gene there and it's functional. There are strains of this pathogen that are what we call Tox minus. So they either don't have the gene for this toxin or it's present there, but it's not functional. So there's some problems. It may not, the sequence may not be fully functional or various other aspects that prevent that toxin from being produced or from being functional. And if you look through the scientific literature, it was thought that these Tox minus strains of halo blight were not important in terms of causing disease or losses. However, in 2016, when we showed that there were Tox minus strains of the halo blight pathogen present in some of these bean seed crops, they were highly virulent. They caused pretty severe symptoms, as you can see on the lower right-hand photos, where there's no halo, but very, very severe symptoms on these leaves compared to the strains with the halo on the left that were our control strains to test. And so this is, I think, a helpful comparison for those of you sort of thinking about symptoms. So these were leaves we took off plants we inoculated in the greenhouse with different strains of Pseudomonas. So the top left-hand one is a strain of Pseudomonas syringae that goes to soybean, not to regular bean. So that's glycinia. And you can see when you inoculate regular beans, green beans with glycinia, you don't get any symptoms. So this is not able to cause disease on regular beans. It does on soybeans. When you inoculate with a halo blight pathogen, Pseudomonas aeruginosa phasiolicola, that has a functional toxin gene, you can see the classic symptoms. And here in the middle, this leaf is inoculated with an atoxigenic or tox minus strain of the halo blight pathogen. You can see a lot of spots on here, but no distinct halos. And this picture, fourth from the left, is showing symptoms caused by the brown spot pathogen, Pseudomonas aeruginosa pathophyllus aeruginosa. And here on the right is a check leaf with no symptoms. It wasn't inoculated. And just to show you that in terms of the production of that toxin, this is a Petri plate on which each of these little circles basically represents a colony of the strain we put on this Petri plate. So this is a halo blight strain where it is producing the toxin. These PS numbers represent different strains that came off bean seed crops in Washington. In the middle, we have the soybean pathogen. On the bottom left here, we have the brown spot pathogen, which doesn't produce this toxin. So we put a spot of the bacterial strains on this plate, and then we flood the plate with E. coli. E. coli is a really fast-growing bacterium. It's inhibited by the presence of this phaseotoxin. So you can see where the halo blight pathogen is growing that produces the toxin. E. coli is not able to grow. So you get this really nice clear zone of inhibition. And you can see that the soybean pathogen doesn't have any inhibition because it doesn't produce that toxin. Same with the brown spot. And if you look at these PS numbers, 009, there's no clearing around it. So this is a strain that did not produce a toxin. The 07, 05, and 03 produce very limited zones of inhibition. So they have that little bit of a functional toxin, but nothing like it should be if it was a fully functional gene. So this just shows how ways we can differentiate some of these tox plus and tox minus strains. And in terms of looking at the family tree and the relationship of these, this is how we confirm the pathovirus identification, if it's brown spot or halo blight. There's 4 genes we look at and the sequence of these and basically make up a family tree. So this is, you just consider it like a family tree. He has the ancestors and he has all the things that are closely related are more closely together on the fine end on the right-hand side of the tree. So these strains here, 09, 13, 14, 10, 11, 12, you can see they're 2 distinct clusters. So these 3 are closely related, these 3 are closely related, and they're most closely related to brown spot pathogen, Pseudomonas aeruginosa. Whereas these 3 here, 3, 5, and 7 are most closely related, extremely closely related to the halo blight pathogen. And that's one of the ways we confirm the identification. So in 2020, there was a bean seed crop under center pivot irrigation that tested positive. It was confirmed by with both a sample pulled out of the field during production and a sample of the harvested seed tested positive. And it was found to be infected with the brown spot pathogen, as I said, both from the leaves and from harvested seed. And we sent samples to a certified lab that also confirmed this. However, they got a negative seed test result when another seed sample was sent to them. So this, of course, creates some confusion when a grower or company might get contrasting results. But here you can see an example where we did testing in the greenhouse. We take these strains we get off the leaves or the seed, we inoculate plants, and it's pretty distinct when they're pathogenic. One way we can also run a quicker test, because we don't have to wait for beans to get big enough to be able to inoculate, is you can do a bean pod stab. You stab the pod through sideways, not through the suture, but sideways like this, and you can see if it's brown spot or halo blight, you get these very distinct symptoms on the pod that show that it is pathogenic. If it's not pathogenic, you won't get these kinds of symptoms on the pod. And this is, again, just showing some symptoms of inoculated plants in the greenhouse for verifying pathogenicity. Unfortunately, in 2020, there was a lima bean seed crop under center pivot irrigation that tested positive for the brown spot pathogen. Here you can see leaves in the greenhouse. These were plants tested by a lab in California, and they also confirmed it with DNA sequencing and these pathogenicity tests. So in 2019, I started receiving phone calls from many bean growers around the Columbia Basin saying, we've got spots all over my fields. These are both bean seed crops and regular production beans for processing. And in most of these calls, the growers are saying it's pretty much 100% of the field. which is very, very not typical of a disease. So here's a photo I took of samples that came from Walla Walla, samples from the Oregon side of the Columbia Basin, samples from the Warden area, samples from the Georgia area, and all somewhat pretty similar, all of these. And we're talking thousands of acres of production. And so there's a really big question about what was going on, because the uniformity of these symptoms in the affected fields and the widespread distance across the Columbia Basin where these symptoms were showing up is very not typical of a disease, suggests that it was something abiotic, non-pathogenic. And so when I started talking to the folks that were calling about this, as I said, they mentioned that it was pretty much close to 100% incidence in these fields. They appeared very rapidly, the symptoms. They appeared during the hottest time of the season. We'd had conditions over 100 pretty regularly around this time. It was widespread across the Columbia Basin from the Oregon side all the way up to the Quincy area. Thousands of acres. It seemed to be most problematic in lima beans. And as we sort of tried to do some investigation about what had been happening in these fields, is that in almost all of these fields, symptoms started showing up within about a week or so after either aerial or ground tank mix applications of various products. And when I investigated to see what was used in these tank mixes, it was a wide range. And sometimes it was, very often had a copper in there because they're concerned about these bacterial diseases, often mixed with a fungicide because of trying to control white mold, often mixed with the fertilizer, including micronutrients, sometimes with insecticides, often with adjuvants, and other ingredients of various things that people believed were going to make a difference in the bean seed crop. And we think what appears to have happened is that these tank mixes that get quite hot in terms of risk of phytotoxicity, especially on certain types of beans like limas. And when you put them on when the temperatures are over 100, there's a much greater risk of this kind of phytotoxicity. But you can see the concern that these symptoms might have been a quarantine disease. And it did turn out one of these fields actually had brown spot and had to be destroyed, but the others were all phytotoxicity issues. We've seen other symptoms show up in bean seed crops and bean crops in the Columbia Basin over the years that have raised concerns about whether or not they could be these quarantine bacterial pathogens. So this is a photo taken of a field in the central Columbia Basin. The grower got really concerned when his crop started showing these symptoms. Here's some other photos taken by, this was Kerry Waleb, our extension educator for the North Columbia Basin. Just a lot of bronzing and stippling and necrosis and yellowing of the leaves, a wide range of symptoms. And one of the things that we ran into, particularly in years like 2013, 14, and 15, which some of our hottest summers on record, and very, very severe wildfire smoke, is that we started running into basically forms of air pollution or smoke, air pollution injury. Some beans are very sensitive to the pollutants in the air, and you can get basically under these conditions of these tremendous wildfire smoke where you can't even see the sun. You get this happening for days, you get these inversions. We start to see quite a lot of damage from these wildfire smoke related air pollution. Azuki beans seem to be highly sensitive. We saw a lot of Azuki bean crops showing problems. Some cases, the problems were tied to things like spider mites and thrips. So it was really important because in those cases, if it was a quarantine disease, the crop would have to have been destroyed. And if it wasn't, then we needed to clarify that so that growers knew they were okay. It wasn't a risk of infection moving within fields or between fields. So in 2020, unfortunately, this past season, we thought maybe we'd be clear of this, but 6 bean seed crops, all under center pivot in 3 different counties in the Columbia Basin, tested positive for quarantine bacterial pathogens. This included brown spot and halo blight. And what's very interesting is 3 of these fields were planted with the same seed lot that had been grown in Washington over 5 years of periods, and seed held onto and planted again. And under the WSDA bean quarantine, you don't have to test a bean seed lot for these bacterial pathogens if the bean seed was grown in Washington state in the protected area. So they hadn't been a history of testing these lots when they went from generation to generation because they were continuing to be planted in Washington state. And yet in this 5th year, that seed lot was planted, 3 out of 5 fields planted with that seed lot tested positive. And we confirmed the identity of these strains with DNA sequence and pathogenicity tests. Excuse me, as shown here with a bean pod test, which we can get results in about 5 days, as opposed to weeks to get the foliar path test results back. And here's the DNA finger tree showing strains that matched the halo blight pathogen and strains that matched the brown spot pathogen from bean seed crops grown in Washington. All right, Steve, time for a poll question.
All right, Jason, you want to run that poll question? The timestamp on this one is 10:32. 10:32.
Oh, this one's a race. Are we going to get the answers to these questions?
Okay, about 5 more. Okay, Jason, I think we're probably there.
Great.
Yeah, good question. As I understand it in Idaho, unless the soybean seed has passed the phytosanitary inspection by either crop improvement or ISDA, it is not allowed to be grown in Idaho, although we do see some come in on cover crop mixes.
There's a 50-50 answer to that one.
Yep.
Great. All right, so let me proceed. Okay, so, how do we manage these bacterial diseases in bean seed crops? It's really been disconcerting to bean seed growers when they start to see the regularity with which we're seeing these quarantine pathogens show up. Growers obviously are really concerned about the crop loss. Seed companies are concerned about the crop loss, that it's not sustainable. And how do we begin to get a better hold on these and do a better job of preventing these outbreaks? So obviously, of course, the first thing that's required under WSDA's bean seed quarantine is that you're supposed to use certified pathogen-free seed produced in semi-arid regions, because seed produced in more humid climates tends to have a higher risk of becoming infected. There is the ability to treat with antibiotics like streptomycin. I'm not sure what's registered for use on beans in Idaho, so I apologize if this is not in place. So make sure you read the label and follow your state's laws. There are some resistant cultivars to certain races of some of these pathogens, but the problem is, as a bean seed grower, you get a contract to grow a particular variety, and so you don't get a choice on this. And very often we don't know what races are necessarily present in an area. Because these pathogens are not seed-borne, not soil-borne, it's really important that you rotate, use a crop rotation of a minimum of 3 years, so that you get full breakdown of those residues, and the pathogen inoculum in the soil will die off as those residues are broken down. Eliminating weed hosts of volunteer beans is really important because they provide a green bridge from season to season. There's a big question about using furrow or rill irrigation versus or drip versus overhead, because there's less splashing, and that reduces the chance of spread. In fact, one of our bean seed growers in Washington State who had an outbreak in his field and lost his crop the subsequent year, he tried raising his bean seed crop on drip irrigation. And from what I could tell when I visited the field, it looked fantastic. I'm not sure if it's economically viable because it's a substantial increase in cost, but he had a very good looking bean seed crop under drip irrigation. The theoretical recommendation is to limit both personnel and equipment moving through the crop when the canopy is wet. And obviously this sometimes is just not feasible logistically because of everything that needs to be done. But when a canopy is wet, there's more risk of these bacteria increasing on the surface of the leaves and potentially being moved around. One of the key comments, as we've had discussions with State Department of Ag inspectors, is making sure that they're wearing sort of at least knee-high boots or protective material over their shoes and their legs, so that when they leave a field, they can disinfect that material and not carry an inoculum from field to field. Copper-based foliar spray programs are used quite widely in the Columbia Basin, and I anticipate that's probably somewhat true in Idaho as well. I think one of the really, really important parts for growers to recognize about coppers is that they're not systemic, they're purely protectant. They have to be used preventatively, so they don't cure an existing infection. And that's one of the difficulties is coppers are not a silver bullet to control bacterial diseases. They provide limited protection, and that's, we really don't have good systemic fungicide, bactericide program for these diseases. Unlike, for example, if you're dealing with white mold, there's some pretty good systemic fungicides, but we don't have that for the bacterial pathogens. So spray programs have limited efficacy. As I mentioned before, managing crop residues is extremely important. If you leave a lot of crop residues on the soil surface, if it's an infected crop, that pathogen is going to persist longer. And I don't mean to contradict the no-till video that went on and the promotion of no-till production of beans. It's a very good practice. It's just that there can be a conflict in cases where you do have one of these quarantine diseases show up, because residues on the soil surface are going to break down much slower than residues in the soils. Because I've mentioned pesticides, this is just a disclaimer slide so you don't sue me or WSU if you use a pesticide inappropriately. It is your responsibility to follow the label. Okay, so one of the discussions that we've been having with the bean seed industry is how do we learn from what's happened over the last few years that these diseases have showed up, and how can we move forward in improving our ability to manage these? When you look at the incidence of infection in these fields that have tested positive and the distribution of the symptomatic plants, there's quite a wide range. I've showed you that one photo of the cranberry bean seed crop where there was just a whole bunch of hot spots throughout the field, which clearly indicated that the original seed lot planted probably had a fairly high incidence of infection. But in many of these fields that have tested positive, there's only been one or two or a very limited number of hotspots. And one grower mentioned he had 2 pickup truck size hotspots in his field. And that potentially could have resulted from 2 infected seed out of the entire seed lot planted over his 100+ acre circle. And so it's a numbers game in terms of how many seed might be infected in the seed lot that's planted. If you suspect you have bacterial disease in your field, we did run into some problems with samples being shipped to labs that didn't have the capacity to do the full rigorous tests. Sometimes samples weren't sent overnight, they were shipped on a weekend, and they got, in 100-degree weather and conditions when that sample arrived at a lab were pretty deteriorated and hard to work with. Obviously in Idaho, you have the Idaho State Department of Ag Seed Lab that Liz Vavrika runs, which is fantastic, excellent resource. There are commercial labs that can offer testing for these pathogens. Eurofins in Colorado, Iowa State University's lab in Iowa, Ames, Iowa, and California Seed and Plant Lab in Central California are labs that are have gone through auditing and certification for offering these tests. One of the difficulties of testing for these pathogens is that there are standardized assays for testing bean seed for halo blight and common blight pathogens, but there isn't a standardized assay for bacterial brown spot, and neither is there for bacterial wilt. And so that's one of the questions that have been talked about, is the need to develop standardized assays for all of these pathogens. And when a lab does say, isolate or say that, yes, the sample tested positive for one of these pathogens, it's really important that that lab follow up with pathogenicity tests and DNA fingerprinting to make sure that the identification is correct, because there are a lot of very closely related bacteria. Pseudomonas syringae is extremely abundant everywhere. My technician jokes about being able to isolate Pseudomonas syringae from a doorknob if you try hard enough. So making sure that you know which pathovar within Pseudomonas syringae is there, if it is a Pseudomonas syringae organism that comes out. So, we've 18+ fields in Washington infected since 2009, and apparently 0 that we know of from 1968 to 2008. That's a pretty big change in circumstances. One of the things we know that's happened in the Columbia Basin is we've had a significant increase in the amount of production with overhead irrigation, as the amount of rural irrigation is decreased very dramatically in our area, and that certainly does increase the risk of bacterial diseases. Another loophole that perhaps we need to close or think about is that stock seed is stock seed being tested effectively? Are stock seed lots all being tested under the current quarantine in Washington? As I said, you don't have to test a stock seed lot if it was grown in the Columbia Basin, and that we know in some cases has actually been the source of infection is the stock seed lot that was grown in Washington state. So the seed industry is working with WCA to consider not allowing that to happen. So as I mentioned, in halo blight, these are the years that halo blight has shown up in Washington state. Common blight only 1 year, 2009, common bacterial blight, but brown spot, again, quite frequently, just to show you the frequency with which we now are having to deal with these pathogens. So in 2019, one of the companies that ended up losing a seed crop to one of these diseases approached me about getting their stock seed lots tested. And my lab is a research lab, not a commercial seed testing lab. So I don't have the capacity to just run through a bunch of seed lots. So I recommended that companies send their stock seed lots to one of these commercial seed testing labs that's certified and audited. And they did so. And what was quite eye-opening is that 11 of these stock seed lots tested positive for one or more bacterial pathogens. That's a lot. That's not where we want to be. We need to be in a better state of that. So 8 of those seed lots, we received subsamples at my lab, and we tested them as well to see if we got consistent results. And subsamples of the same 8 lots was sent to a second commercial lab to see how those results came back. And this was kind of a summary, there's a lot you had to consider, but basically the columns represent the different pathogens that were tested for in those seed lots. The lots here 1 through 8, my lab WSU, and then lab X and lab Y, just for anonymity's sake. But you can see that in some cases, like seed lot 1, lab X picked it up at 10 to the 5 CFUs for Xanthomonas, which is a pretty high amount of infection, but we did not pick it up in our lab, and neither did lab Y. In some cases, like seed lot 2, lab X picked it up at extremely low level. This is a very, very low level infection. My lab and lab Y didn't pick it up. In other cases, like seed lot 5, all 3 of the labs picked up this pathogen. And one of the issues you run into is when the number of bacteria picked up is very, very low, like seed lot 7 here for can't see the pathogen because I've got the bar is blocking the header, but very low level like 3.8, or here 20 CFUs. This may be a case of a seed lot having a very, very low incidence of seed infected, very few seed actually infected. And therefore, when you pull a sample to test, it's very likely that you could get differences among labs because of the very low incidence of infection in a seed lot. But when you get a high level infection, like here, seed lot 8 tested positive for 2 of these pathogens, when you're getting levels like 10 to the 6, every lab should be coming back with a positive, because that's a very high level of infection. So this shows some of the issues of discrepancies among labs, and what do you do with that? And the fact that, look at how many of these seed lots tested positive for bacterial wilt, 1, 2, 3, 4, 5, 6 out of 8, depending on the lab that ran the test. So what is going on with bacterial wilt? Historically, we haven't really had to think too much about this disease. Where's this pathogen coming from? How does it behave? So it's a very different bacterium than the Pseudomonads and Xanthomonads. It's Cotobacterium. There's different species of Cotobacterium and different pathivores within Flockham fascians. And this pathogen actually gets into the vascular tissue, gets into the xylem and causes a wilt. So it looks more like a wilt than it does a leaf spot pathogen. And it infects a range of legumes, not just beans, but also soybean, pea, and Begna, others. It was first reported in the US in 1922 in South Dakota. That was the first time it was reported anywhere in the world. In the 60s and 70s, it spread very fast through the Central Plains and Midwest of the US, up into Central Canada and in Mexico. So it became quite widespread. And then the appearance of this bacterial pathogen all but disappeared until about the early 2000s. And then we started to see the prevalence of bacterial wilt increasing again in these same areas, the plains and the Midwest. And it's now widely distributed in the USA and in some other countries. It is considered a quarantine pest in the European Union. And so this is a concern if bean seed is going to Europe from this area. It is readily seed-borne and seed-transmitted, particularly because it causes a wilt type pathogen. The symptoms, as I said, are quite different. You get the leaves turning, droopy, wilting. You get this chlorosis and necrosis between the veins. We call this firing, stenting, death, and reduced yield. The seed can be discolored, shriveled, or asymptomatic. The bacterium itself, this is what's very interesting. This picture in the top right-hand corner, these bacteria can come in different colors. They're all the same pathogen. They all cause bacterial wilt, and yet they can come in different colors. So it's also called the bacterium of many colors, which is very unusual for these bacterial pathogens. It does best under high temperatures, so it's a warm season pathogen. Unfortunately, it can survive in seed for a very long time. There's records of it surviving in seed for more than 20 years, and it survives in infected crop debris. What really complicates the diagnosis is that there's evidence that there's strains of CFF, for short, that are pathogenic on beans and strains that are not pathogenic on beans. And how do you differentiate those? And perhaps more disturbing is there's some publications coming out in the last few years showing that these strains of CFF can also colonize other hosts that we know are grown in rotation with beans like corn, sunflower, potato, canola, sugar beet. And so what role do these other hosts play in keeping that inoculum around from season to season, providing a green bridge for bean seed production? So where do we go now? We're clearly facing a concerning situation with these bacteria becoming more frequent in occurrence in bean seed crops. We know that there's evidence infected stock seed lots are getting planted in the Northwest despite testing, or perhaps because they're not getting tested. We know that the seed health tests are destructive. You sample, you destroy that seed in the process of testing, so you can never fully prove that a seed lot is negative because you'd have to destroy the whole seed lot. And the distribution of the pathogen in seed lots, these pathogens, is very uneven. So it's an issue of sampling error. And the probability of picking it up varies depending on how uniform it is in a seed lot, how high the level of infection is, also what field conditions are like. If conditions are particularly dry and the type of irrigation you have, you may not pick it up if it is there. There's variation in protocols for testing among labs that we discovered. There's also variation in technical expertise, experience with these pathogens that can influence what the results turn out to be. And some really important considerations about understanding the seed assay, the seed testing protocols, the limitations, the interpretations, the financial implications of false positives and negatives is really huge because these are crop destruction requirements. And then this question about irrigation. Should bean seed crops be allowed to be grown under overhead irrigation? If we didn't allow that, it would really create constraints around the capacity to produce bean seed crops in Washington because so much of the production, most growers are now using overhead irrigation. One of the really interesting questions that's come up is this aspect of moving equipment between crops. So we have a lot of contracting services in the Columbia Basin, and I suspect in Idaho too, based on some of the questions earlier on with the discussion around the videos, where you get contractors who may do contract swathing or may do contract spraying. And you have equipment being moved among hundreds of fields of beans because you're doing a similar spray program, for example. And what's the risk that that equipment might potentially move these bacteria if the equipment is running through fields when it's after rains or after irrigation and potentially picking up inoculum? And should we be requiring testing of stock seed lots even if they're produced within the protected area? So this, in red here, we've got some bullet points that the seed industry and the Seed Growers Association in Washington State are considering. So reassessing the seed health assay protocols, looking at ways of trying to develop greater proficiency in the labs that do offer testing to make sure we all get similar results if we get similar samples. And using there's a organization in Europe called Nachtenbau, which basically offers certification of labs in the EU. And they require that those labs get basically a proficiency test run every 3 years using blind seed samples to make sure that they're still able to offer tests with reliable results. And I think that would be a useful example we could incorporate in the US. So what resources do you in Idaho have? Liz Vabrica, the state plant pathologist, is a fantastic plant pathologist, very experienced with these pathogens. You're very fortunate to have Liz serving you. I couldn't find a picture of her, so sorry, Liz, this is you. At least it's a happy onion. Another resource for you is James Woodhall. He's a assistant professor at the University of Idaho. He's received a specialty crop block grant from the Idaho State Department of Agriculture to look at testing methods for all of these pathogens that can be a concern in bean seed production. So he's evaluating the existing methods and looking at develop improved DNA testing methods. So potentially reduce the seed testing time to less than 24 hours versus multiple weeks for confirmation, both seed testing and folio samples. He's also wanting to use these to monitor the pathogens in fields using these things called JB traps, so basically funnels that trap any water that arrives in the fields into these membranes that he can then test. And he'll be doing this testing in combination with Liz's ISDA lab for comparison of methods. And if you'd like to get hold of James to participate in this study that's funded by the ISDA Specialty Crop Block Grant, here's his email address. And so with that, oh, I think I have a couple more slides. Sorry, Steve. Just a reminder that these bacterial pathogens can spread very rapidly. They don't discriminate between seed crops and production crops. And so one of the big questions for us, certainly in the Columbia Basin, and I pose this question for Idaho as well, is our seed growers are surrounded by a tremendous amount of acreage of processing bean crops and other bean crops. And so the success of the bean industry with respect to controlling these pathogens really is dependent on cooperation among seed growers and production growers, not just the seed growers. Seed crops get inspected 3 times a season in Washington state. The production crops, the processing crops and dry bean crops don't get inspected at all. There's a heck of a lot of folks that visit these other crops besides bean seed crops that could serve as unofficial sort of inspectors for monitoring the risk, submitting samples to diagnostic labs, helping implement management practices. For seed growers, it's important to consider not planting seed crops in proximity to where the disease showed up a previous year. And I think really considering this issue of equipment moving between fields to try and limit the risk of spread into seed crops. Laurel Skogsberg from H.M. Klaus recently, at the request of seed companies that have been affected by these diseases, has started these bean bacterial disease working groups. So there's 3 working groups. There's one that's looking at rules and regulations across the tri-state region to see if modifications are needed to help improve the protection of seed crops. Chet Kurowski is looking at, has a group he's leading to look at cultural best practices and in-field treatments. Are we doing the best thing we can? Do we need to kind of brush up on best practices to limit the risk in fields? And I'm working with a group to examine the pathology, testing and detection technology because of some of the issues we run into. So really, these 3 groups are looking at how do we come up with recommendations for improving our ability to produce clean bean seed in the Pacific Northwest. And so with that, Steve, I'll end here. This is the last slide, and I'm not sure if there's time for questions.
Yeah, we have a few questions. We'll take yours since I know that you're on kind of a time crunch this morning. So we had 3 questions in the question and answer. The first one, have you ever seen an increase in bacterial infection after a fungicide application?
Really good question. And Clendon, thank you for that question. I haven't, with respect to beans, I haven't done any analyses on this, but I will tell you that in other crops I work on with bacterial pathogens in particular, I'll mention an example with carrots. So we've done a lot of work with bacterial blight in carrot seed production. Again, it's one of these seed-borne pathogens. It's a big concern for the carrot seed industry, and our growers have struggled with this. And we have shown during some studies, some trials we did, that when you apply a fungicide, in this case, we were looking at a product called Tanos, which growers were saying, well, it has some bacterial activity. Can it improve our ability to control these bacteria when we put Tanos on with coppers? And we showed that when you put Tanos on, you actually can make these bacterial diseases worse. And what happens is when you put on a really good fungicide like Tanos, which has 2 active ingredients, you kill a lot of the what we call the epiphytic, the naturally occurring fungi that exist on the surface of the leaf. And there's less competition for the bacterial pathogen if it lands or when it lands on that leaf. And so it can colonize much more rapidly and build up in greater populations. And we showed it very, very clearly in some studies we did that you can actually exacerbate a bacterial disease problem by wiping out the fungi that might occur naturally on a leaf. So yes, there is this unfortunate consequence. That doesn't mean we shouldn't spray fungicides. Obviously, if you need to manage diseases like white mold, it presents a bit of a dilemma that these fungicides are controlling fungi, which actually can compete with the bacteria for the ability to colonize and establish in a crop. So this is a dilemma we have to think about very seriously, and it makes you wonder when these tank mixes get put on that include coppers and fungicides, are we kind of contradicting what we're trying to achieve with some of these heavy tank mixes? So very important question, Clinton.
Okay, the next, is Adzuki bean regulated the same as Phaseolus vulgaris in Washington and in Idaho?
So in Washington State, if it's a seed crop, a zucchini bean seed crop, yes, it's regulated the same way as Phaseolus vulgaris. I'm not sure about Idaho. I don't know if there's anyone on the call from ISDA that can answer that question. I apologize. I don't know the details, but I suspect it is in Idaho, given what we have with WSDA's rule. If it's a seed crop, it gets regulated the same way.
Okay. I'm promoting Kathy with Idaho Crop Improvement Association to panelist. Kathy, can you answer that question for us for Idaho?
I can. Azuki beans also fall under the regulated diseases in Idaho. So.
Thanks, Kathy.
Okay, great. Thank you. And the third question, what do you think is most important, presence of inoculum or susceptible host?
Yeah, that's a tough question, Clendon. To have disease, we always talk about the disease triangle. To have a disease show up in a field, you've got to have the pathogen, you've got to have the host, and you've got to have the right conditions environmentally. And if one of those three is missing, you don't have disease. So if you have the pathogen but no host, you're not going to get disease. If you have the susceptible host but no pathogen, you're not going to get disease. If you have both but you have very unfavorable conditions, you won't get disease or you'll get less disease. And thinking about the situation where historically we haven't had these problems show up very much, at least since the 70s when they were an issue, why are we starting to see these? I think the issue here is that we're seeing inoculum come into these seed production areas that historically we've been able to keep it out of. So, we have to, in this case, susceptible host is the seed crop or the bean crop. You've got to have these crops. We want to be able to produce bean seed in this area. The reason bean seed production is concentrated in the Northwest is it's semi-arid. Arid conditions are not favorable for bacteria. Unfortunately, we do create more conducive conditions with things like irrigation. And so I think in the situation we're dealing with, inoculum is the question we're having to try and manage, Clendon, that we're introducing inoculum somehow. Now, I do believe that inoculum's coming in on stock seed based on the patterns we've seen in these fields. And the question is, why is that happening? How is it happening? Some of it we know is related to the fact that you can get very, very low levels on seed. Some of it could be that it's coming from areas where these pathogens are prevalent and the seed's being tested perhaps at a lab that doesn't have full proficiency, or that it's at such a low level it's not being picked up. I've heard all kinds of comments as we've investigated these cases where the disease has shown up. Seed maybe came from the Midwest. What's happening with the fact that seed could be coming in these processing bean crops if it's not being tested, and potentially open up the risk of movement to a seed crop. This past year, one of the seed companies called me up and said, can I ship over to you 2 bean plants? Can I ship 2 bean plants to you that I picked off the header of a swather that had come out of a dry bean crop and was moving into one of his bean seed crops. And he stopped the equipment from coming into his field because he saw these 2 plants hanging off the swather. And basically they tested positive for brown spot. So this piece of equipment was actually moving 2 plants that were attached to the header into a bean seed crop. And he stopped the swather before it came into his bean seed crop. And we tested the plant and they were positive for brown spot. Now that doesn't mean necessarily that since it was at the swathing stage, what's the risk that it could have got into there? That those plants shouldn't have been on there anyway, because you don't want seed from a different variety getting into that seed crop, but it just really highlighted some of the risks we face that we may not be paying as much attention to. And that was one of the things that the cultural working group, the cultural practices working group that Chet Kurowski is leading, is going back and saying, have we gotten too complacent? Have we gotten too satisfied, not aware of the practices we need to be more strict about to keep the risk of inoculum out? So sorry, long-winded answer to that question, but I think it is a case of trying to keep the pathogens out of these production areas.
Okay, thank you. I don't see any additional questions at this time. Kathy, quickly, in a related question, came up a little bit earlier about the legality of soybeans in Idaho, and I'm not sure I answered that very authoritatively. Would you please inform us about that?
Okay, so you did really well. You can grow soybeans in Idaho if they are either grown in Idaho originally and inspected and grown as a class of certified seed, or if they are serology tested upon arrival in Idaho and they pass that serology test.
Okay, thank you. Hope that cleared that up a little bit for whoever had asked that question a little bit earlier. Okay, Dr. Dutoit, thank you for that great and very informative presentation. I saw some comments back about how you do a very good job of speaking in a layman's voice so that our producers can kind of understand what you're saying. So we really appreciate that. Appreciate the information for you giving us some of your time this morning.
Thank you. Appreciate the opportunity.
Okay, have a great day. All right, Jason, we're up to some door prizes. So we're up to the second $50
Yeah,
gift
and
card.
we actually, our person that we tried to give that away to did not reach out to us, so we're going to give away that $25 one as well. So
Okay.
the $25 one is going to go to William Casey. If you are here, I think I saw you in the attendees. The $50 one is going to Terry Helms, although I can't confirm whether that's you or not. There is a Terry in the attendees, so if that is you, let us know. So the $50 to Terry Helms, but I need you to confirm in the chat that that is you, Terry. And then the $100 one goes to Camby Reynolds. So any of those of you that have won, please stay so I can get your information in the chat so we can get that taken care of and send you the gift card. We also have a survey. If you didn't take it yesterday, I will get that in the chat here in just a second.
All right, thank you. The question had come up yesterday, how do you get your credit? When we compile and we link your name to either your poll question or your timestamps, we will send all that to Department of Ag, and you will receive an email from the Department of Ag with today's session number that they have given it, saying that you've received that one credit. So be looking for that from the Department of Ag here within the next, I would say, a couple of weeks. It's going to take us a little bit of time to kind of put all of this stuff together. Okay, thank you. Dr. Toyd had said that she would be happy to share a PDF of the program, and that question had come up a little bit earlier. We will see about getting all the presentations put together and probably housed on the Idaho Bean Commission website, maybe, Andy. And then we'll fire out an email to everybody who registered to let you know that that is there. Okay, again, if you were worried about the poll questions, oh, go ahead and unmute there, Andy.
No, just going to say that sounds good. I can put them on our website for sure. And if anybody wants to email me, I had it on our or on my my talk yesterday. It's bean@bean.idaho.gov, and I can always email those out once I receive them. So.
All righty. Let's see. Rick B raised his hand. Jason, how do we talk to Rick?
You need to hit allow to talk.
All right. All right, Rick. I don't see Rick.
I don't see. He dropped out. He was trying to hit leave, and he hit
Leave
the
probably.
raise
Okay.
hand instead.
Yeah.
So yeah, those of you that won one of the prizes, stick around for just a second, and I'll message you in the chat.
All right. So we had that kind of wraps up our program, and I'd really like to thank our speakers for the last couple of days. Before we all run out, we did offer the opportunity for some additional question and answer with some of our panelists. If you had some additional questions for the panelists today. Let's see. Okay, I see that Rick raised his hand again, but I don't see him there. Maybe you can help me with that a little bit.
Yep, I'm
Okay,
requesting for him to unmute, and he is not, so it might be on accident. But Rick, if you're there and you had something important to say, I guess this is your chance.
Okay, Kathy, it looks like there's maybe a question here for you. Does that include cover crop soybeans?
It does. Absolutely. So again, be careful. Look at those labels when you are sourcing your cover crop seed.
Okay. Let's see. Is there anything else in the chat here? I don't see any questions there. Anybody else have a question for any of our panelists here as we're wrapping up this morning? Again, I'd like to thank them. I'd like to thank the Magic Valley Bean Committee for putting together a great program. Really like to thank Jason for making all the technology happen. Without him, I don't know. Been a little rough. I'd like to thank our producer panelists who joined this morning and answered questions and really helped add some depth to that program, and to Jackie to putting together some great video tour for us. And not seeing any additional questions. Let's see. All right. Oh, sorry. Jason's sending a. So Jason, do we need to leave this hot for the chat?
I'll leave the meeting going, but yeah, it's technically officially over. Other than the winners, I'll talk to them really quick via chat. So thank you for joining us.
Well, with that, thank you everybody for joining us. I think we had a great 2 days and a good program. Please fill out that survey that Jason put in the chat there. That really helps us gather some information on how we did. And with that, everybody have a great day. Thanks again to the producers, and hope that you have a great production season.
Thank you, Steve. Thank you, Jason. Appreciate all your help. Great job.